Firstly, thank you organisers for giving me the opportunity to speak today, and thank you all for attending. I'm Damion, and I'm a research radiochemist and cyclotron facility manager for the Centre for Advanced Imaging at the University of Queensland. Today I'll be talking about our experience with the PET tracer fluorine-18 DPA-714 and our journey in going from manufacture for preclinical use to manufacturing a product that is suitable for use in humans. So let's start with the why. DPA-714 is recognised as a specific ligand for the 18 kilodalton translocator protein, an important biomarker for neuroinflammation. Fluorine-18 substitution in this ligand therefore provides a useful tool for the assessment of various neuroinflammatory responses to disease or injury with PET. That's an area of interest at the Centre for Advanced Imaging. And now the value to radiopharmaceutical science and nuclear medicine. Firstly, the work presented will demonstrate significant innovation in the field of radiopharmaceutical science. I'll show how we have developed a synthesis method that requires much lower quantity of chemical precursor. Secondly, in making this investigational medicinal compound available for clinical researchers, there is now a new tool, at least in Queensland, for the assessment of neuroinflammation using PET. The primary aim of this work was to establish the manufacture of fluorine-18 DPA-714 to the quality where it's fit for use in humans. We had already developed the manufacturing QC methodology suitable for preclinical applications. However, to increase the product quality so that it becomes fit for use in humans was quite a large undertaking and included translation of the production systems from the research lab into the cleanroom setting, to develop and upscale production methodology, establish critical quality attributes based on literature and pharmacopoeial monographs, develop and validate QC test methods, establish microbiological assessment of the product, and to fully validate the manufacturing process. I'll begin with describing our initial work for preclinical studies. We based the radiosynthesis on that developed by Michelle James and co. It was first reported in 2009, but with a few tweaks that came from our work in the production of fluoromethyl tosylate, where there was a focus on the quantity of base used in the synthesis. This facilitated the reduction in the quantity of precious chemical precursor. I'm not going to go into that too much because there's a publication coming sometime soon on this work. So starting with 15 to 20 GBq of fluoride, we would make around 2 to 3 GBq of product using the Synthra automated radiochemistry module with very little precursor, as low as 0.5 milligrams. The advantage of the Synthra module is the ability to perform semi-preparative HPLC relatively easily. Here's one of our semi-preparative chromatograms showing the radio trace, followed by reformulation using SPE cartridges. Quality control was by HPLC. Here's an example of the radio chromatogram on the analytical system, and that's showing greater than 95% radiochemical purity. A typical radiochemical yield was around 14% at end of synthesis, with a good mole activity averaging 185 GBq per micromole. And you can see we did quite a number of productions, over 113 productions. So I'd like to give you a quick overview of the synthesis. We had 2 Synthra modules available in the research lab, both located inside Team hot cells. And these pictures show this particular hardware. Having developed a sequence of events for the automated synthesis, we can see what is happening during a production using the module's graphical user interface. That's it on the screen now. So before we introduced radioactive fluoride from the cyclotron, we set up the module. We had our tosyl precursor. We add that dissolved in solvent, vial A3. We also add all other reagents and consumables, including the quaternary ammonium, methylammonium QMA cartridge and C18 cartridges, to their respective positions. With the QMA on the left side here, C18 on the right side of the diagram. Now, both these cartridges are of low loading, which means they can be eluted with lower mass or volumes of eluent, and that's advantageous, of course. The process begins with the transfer of radioactive fluorine-18 from the cyclotron target to the Synthra V vial here, the collection vial. We then separate the fluoride from the target water using the QMA, which is here. And we then elute the fluorine-18 off the QMA with a solution of cryptofix and carbonate base that's from A1. We push this mixture through into the reaction vessel here. And again, it's the base is the important aspect of facilitating the amount of precursor. The fluoride is now azeotropically dried in the reaction vessel under helium flow and vacuum. The tensile precursor and solvent is added to the reaction vessel, and the radiolabeling is achieved at 100 degrees over 10 minutes. After this, the crude reaction mixture is aspirated with a syringe and then injected onto the HPLC loop, where we then do our semi-preparative purification. With a retention time of around 20 minutes, the product peak is collected and diluted with water in this collection vial. It is then loaded onto the C18 cartridge. After this, we can then wash any residual mobile phase away, any solvents away to waste, and then elute with 0.5 ml of ethanol with a final formulation of 10% ethanol in saline, the total product volume of around 5 ml. The next stage in the project was to establish the upscaled method. We found that we achieved good results with a little more tocile precursor, although still a relatively small amount, at 1 milligram. We sourced a GMP grade supplier for precursor, a company called Pharmasynth. The modified method takes approximately 75 minutes from delivery of fluoride to end of synthesis. So I mentioned before, we've effectively doubled the amount of fluoride to start with, 30 to 40 GBq. We wound up with radiochemical yields around 25% and a molar activity of 150 GBq per micromole. We then incorporated our manufacturing. So in tandem, we developed standard operating procedures, and we incorporated this into our pharmaceutical quality system. We also incorporated sterile product filtration in the process. We established product critical quality attributes, and we developed a comprehensive product quality control. So talking about critical quality attributes, in line with this are the quality release specifications that were set in accordance with pharmacopoeia monographs for other fluorinated radiopharmaceuticals. You will notice specifications for molar activity, chemical purity via HPLC, and other standard tests for pH and the cryptofix or the aminopolyether. Typical to parametric release, we then have the retrospective tests, and these are the tests that can be performed in a suitable time frame before product expiry or during the window of time that the product is useful. The specifications here are for parameters such as residual solvents, sterility, apyrogenicity, tests that may take some time to complete. So now that we've established the upscale methodology, we need to make some changes to the equipment to provide for aseptic production, in particular moving the Synthra module into the clean room environment. There was significant preparation, including removal of a different synthesis module. You may notice the IBA Synthera module in the first picture. We're no longer using that. And then modification of the hot cell to make space for the physically larger Synthra module. So you can see my colleague Chris here removing the internal shielding compartment from the hot cell. And finally, we have space to accommodate the Synthra module. In the picture on the right, you will see we've managed to include a dose calibrator in the hot cell and have installed a PVC floor in the hot cell to ensure a flat, even cleanable work zone. So the results of this, for the Synthra module, installation, we managed to fit the module into the hot cell. It was quite a tight fit, get everything up and running. It's really a tight squeeze to get all the components to fit into this particular hot cell. Further to this, we installed a dedicated product transfer line from the synthesis hot cell into the adjacent dispensing hot cell, where the final product is sterile filtered and then dispensed into the sterile vials as required. After implementing a cleaning regime, we had testing performed by an independent NATA accredited contractor to grade the clean areas as per the PICS code and ISO standards. This is predominantly governed by the number of viable and non-viable particles measured, along with the number of colony-forming units found after microbiological sampling. The classification results gave a grade D for the clean room, grade B for the synthesis hot cell, and grade A for the product dispensing hot cell. And that's what we set out to achieve. So having everything set up, including the upscale methodology and SOPs in place, we went about our validation, of our manufacture. We performed 4 productions, starting with between 30 to 40 GBQ for an 18. Again, it's about double that used previously for the preclinical productions. We achieved around 18% radiochemical yields. This is slightly down from the development stage, but we can account for the losses because we added the sterile filter, so you get losses, volume losses in this filtration step. The mole activity is good at around 144 GBQ per micromole, and all the batches passed all QC, release, and retrospective tests. So in conclusion, we're now making F18 DPA-714 fit for use in humans at CAI, ready for clinical researchers. So I wish to acknowledge the work that you did earlier on, and I wish to thank my current team, Louis and Christoph, for their outstanding work. And finally, thank you for your attention.